All right, welcome back. So this third lecture for the histology of the male reproductive system. In this lecture, we're going to be covering the ducts, the epididymis, and the vas deferens. So first, the ductal system. It's a system of ducts that carry sperm from the seminiferous tubules, where spermatogenesis occurs, up to the urethra for ejaculation. There's two sets of ducts, the intratesticular ducts and then the extratesticular ducts. So the intratesticular ducts, just as its name, they begin here in the testicles. Here you have these coils, which are the seminiferous tubules, if you remember. And then these empty via the tubuli recti, which are these straight-looking tubules like this. Tubuli recti. And you can see there are all these straight-looking guys right here. And then they empty into this region here, which is called the Rete testes. And then the Rete testes, after the sperm has left the Rete testes, it actually exits the testicles into the extratesticular ducts. So it goes, you can kind of see it here where it's labeled, these tubules that are all leaving the testicle like this. These are what are called the ductuli efferentes, or the efferent ductules. And they empty into the ductus epididymis, which is this kind of large coiled region right here. It kind of comes all the way down here. It has the head, a body, and a tail down here. You can see that here. And then the tail of the epididymis connects with the ductus deferens, or the vas deferens, which is the last portion of this tubule system. And the vas deferens essentially is responsible for carrying the sperm from the testicles all the way up to the urethra. It travels up through the pelvis and the abdomen, and we'll talk more about that in a few slides. And so you connect the ejaculatory duct that receives the secretions from the accessory sex glands like the prostate gland, seminal vesicle, and it joins with the vas deferens to bring the sperm and these secretions together, and they all join at the urethra. And we'll go through that anatomy in a few slides here. So first, the tubuli recti, as I kind of mentioned on the previous slide, they're these short, straight tubules that drain the seminiferous tubules into the Rete testes. So they're all these little straight-looking tubules like this, very short. Spermatogenesis does not occur in these tubules. So spermatogenesis occurs in the seminiferous tubules. After the sperm have left the seminiferous tubules, they'll travel these to kind of begin their journey up the ductus deferens. The tubuli recti are lined by Sertoli cells, just like how you found in the seminiferous tubules. Then you have the Rete testes, which is this region right here. It kind of receives sperm from all the seminiferous tubules via the tubuli recti, as we talked about in the previous slide. So it comes in here in the Rete testes. And this is a complex network of interconnecting channels, and they're located within the mediastinum of the testis. So this region right here would be the mediastinum. They are lined by cuboidal or low columnar epithelium with a primary cilium. And then they are surrounded by a highly vascular connective tissue. So it makes sense. You need connective tissue to support this area, where an important process of carrying sperm that's newly generated out of the testicles to be transported. And then you also want to have that highly vascular as well to get plenty of perfusion and nutrition. So this is a section, and we'll go through this more in the lab video, but just to give you an idea what we're talking about. So this region all here would be the Rete testes, or the mediastinum of the testes. You can see this very dense connective tissue. It stains pink here like this. And then these channels here are what we were talking about, those channels that comprise the Rete testes. And so if we zoom in here, you can see this simple cuboidal epithelium. If you kind of look at here, it's one layer of epithelial cells, and they take on a cuboidal shape. And so again, within these passages here, or in these channels, you have the passage of spermatozoa from the tubuli recti, carrying from the seminiferous tubules to bring sperm through this Rete testes, and then eventually exiting through the efferent ductules, which we'll talk about in a second, to carry the sperm out of the testicle. So then now we have the efferent ductules, which are this region right here. Again, in kind of a shorter region. So you had the seminiferous tubules here, we'll just say ST. You have the tubuli recti here, we'll just call that TR. You have the Rete testes here, which is we'll call RT, which is this whole region here, and it kind of ends here. And then here you have the efferent ductules. You can see it says efferent back here, but we'll just make that more clear. And so the efferent ductules, they are the first tubules outside of the testicles, and they form the head of the epididymis. So they're carrying the spermatozoa from the Rete testes here and joining here with the head of the epididymis right here. Histologically, the efferent ductules are lined by pseudostratified columnar epithelium. They contain cilia to transport non-motile sperm. They also absorb the majority of the fluid that is secreted by the seminiferous tubules. And the purpose of that is to help further mature the sperm and help them move more efficiently down the ductal tract. There's also a thin layer of smooth muscle that encircles the epithelium to aid in the transport of sperm as well. So this is a great histological section of the efferent ductules. One thing to note is you'll notice a lot of invaginations. You can see that here and here and here and all over here. And you can see it throughout this section here. The purpose of that is because, well, one, structurally it's due to the cuboidal cells that are interspersed among these columnar cells, or this pseudostratified columnar epithelium, meaning that the cells are different sizes and shapes, and so it allows for when you have these cuboidal cells that are interspersed among them, it helps invaginate the epithelium further in. And again, like we said, most of the fluid released from the seminiferous tubules is reabsorbed by the non-ciliated cells of the efferent ductules. And then the columnar cells, with the stereocilia, help move the non-motile sperm along this tract and move it along into the epididymis. And so you can see this is a collection. This portion here in the lumen here is a collection of sperm like this, and you can appreciate the intersperse of columnar. It depends what text you read. Some will say it's a pseudostratified columnar, and then others say it's columnar interspersed with cuboidal. The point is that as you can see, you have these varying invaginations due to the inconsistency of size of the epithelial cells within this part of the ductal system. So the next structure in the duct system is the epididymis. We've already made mention of this already. But again, just to give you an idea of how convoluted and folded the epididymis is, if you were to pull it out, it would actually be 4 to 6 meters in length. It's a significantly folded structure. Again, like I've mentioned, it's the junction between the efferent ductules, which is this region down here, these ductules leaving the Rete testes, leaving the testicle here, and the Vas deferens as well. So you have the epididymis then transitions into the vas down here. And again, like I've mentioned before, it's divided into the head region, which you can see here. This is the head here. This long portion here. Is the body region. And then down here is the tail, which then junctions with the vas deferens. Epithelium-wise, it's lined by pseudostratified columnar epithelium. It has associated loose connective tissue beneath it. We'll show you that in a few slides here. So the primary function of the epididymis is really twofold. It's to absorb any remaining seminiferous tubule fluid that is still hanging around. I know that after the sperm has left the seminiferous tubules, and we mentioned along the way, along this duct system, that the fluid is being reabsorbed, but kind of the last bit of that is absorbed in the epididymis. And then it also serves as a storage reservoir for sperm. It can store viable sperm for up to 2 to 3 weeks. The epithelium itself is composed of the principal cells with stereocilia, and then the round basal stem cells that help regenerate the epithelium. Specifically, the principal cells are also involved with absorbing the fluid, like I mentioned. They also secrete glycoproteins and glycolipids. Those are involved in a process called decapacitation, where they attach to the plasma membrane of the sperm. I'll talk more about that in a slide here in a second. The epithelium also contains lymphocytes that are called halo cells. During ejaculation, there's rhythmic contractions of smooth muscle layers that surround the epithelium of the epididymis, and this helps propel the sperm along the epididymis and eventually into the vas deferens. Now, as far as the muscle layer, the nature of it, how many layers, it actually changes throughout. So in the more proximal regions, when you get into the head region, it contains a thin circular layer of smooth muscle. And then as you progress down here into the body, you have an inner circular and an outer layer as well. And then when you get down into this distal portion here in the tail that forms a junction with the vas deferens here, you actually have 3 layers of smooth muscle. And so you have an inner longitudinal, a middle circular, and an outer longitudinal. And you'll see in the vas, you also have 3 layers that are similar to this. In the vas, you also have an inner longitudinal, middle circular, and an outer longitudinal. We'll show that to you in a few slides. As the sperm passes through the epididymis, it acquires motility, and then also it undergoes that process I was talking about called decapacitation. So this is where their plasma membrane acquires glycoconjugate molecules, those glycoproteins and glycolipids that I mentioned that are secreted by the principal cells of the epididymis. And these glycoconjugate molecules, they prevent the ability of the sperm to fertilize an oocyte. So at this point, it actually isn't able to fertilize an oocyte. It's not until it's released into the female reproductive tract that it undergoes a process called capacitation, where you have dissociation of these glycoconjugate molecules from the sperm plasma membrane within the female reproductive tract, and then at that point, then it's able to fertilize an oocyte. So this is a great histology slide of the epididymis. You can see very clearly this pseudostratified epithelium here. You can see in classic pseudostratified form, it has that kind of stratified look to it in some areas and then not as much. Remember, this is a single layer of epithelium. It's not a true stratified, hence the name pseudostratified. And then if you recall, you have this stereocilia, which you can see here. Here's some sperm and other secretion content within the lumen. And then actually here's a good look at a basal cell here. As you can see, it's in the name, it's in the base and helps regenerate, serves as a stem cell to regenerate the epithelium. And then you have the principal cells like this. And again, here's a zoomed in view of both the types of cells. So the white arrow here in the left picture here is showing the principal cell here, this long cell here with a stereocilia projecting into the lumen. And then the arrowhead here again indicates a basal cell that you can see here. And then also, if you can appreciate, here's that kind of loose looking connective tissue beneath the epithelium as well. So the last portion here of the duct system is the vas deferens, or the ductus deferens. And we've mentioned this already a number of times, but again, it's a long muscular tube, courses through the spermatic cord into the abdomen and then the pelvis to eventually connect the testes to the ejaculatory duct. So it's the conduit for sperm from the testicles to the urethra for eventually ejaculation. The vas deferens is lined by pseudostratified columnar epithelium. It encloses a folded and narrow lumen, and we'll show you this on a slide in a couple slides here. You'll get to see what I'm talking about here. It's also surrounded by loose connective tissue, and then beyond the loose connective tissue, you have 3 layers of very thick smooth muscle. You'll see it on the slide in a second here that the bulk of the structure is smooth muscle tissue. So you have the inner longitudinal layer, the middle circular layer, and then the outer longitudinal layer. And so all 3 of these layers of fibers are actually at a 90-degree angle or orthogonal to each other. So these are traveling 90 degrees to this and 90 degrees to this and so on. And we'll show you that in a second. The thick smooth muscle layer contracts with significant force to propel the sperm during ejaculation. So again, just to review here, you have the seminiferous tubules. Sperm is produced via spermatogenesis. You have the tubuli recti, which are these short, straight tubules like this. They connect with the Rete testes. Then you exit the testicle via the efferent ductules into the epididymis here. And then eventually you make your way through the epididymis into the vas deferens, which is then going to exit here and enter the spermatic cord, which I'll show you the anatomy of that in a second. So here, if you're taking an anatomy course at the same time, this could be very useful to you. If you're only taking a histology course, this content may not be on your exam, but it also is very helpful with kind of understanding the path of the spermatic cord and the function of it as well. And so if you look at it here, you have the spermatic cord as labeled here. This is kind of the outer portion here before you actually cut in and look inside and see what the contents are. So you have the vas deferens here. The contents of the spermatic cord, again, a histology course may not necessarily ask this on an exam, but definitely on an anatomy exam, you would get asked this. So you have the vas deferens, you have the testicular artery, the pampiniform venous plexus. We had talked about that. Remember, it cools the blood in the testicular artery. And then you have the cremaster muscle as well. So you can see the vas right here. Point some lines here to point it out to you. You see it's this nice strong tube here going through, and it's exiting, like I said, from the testicle and ascending upward this way. Once you get up here, this is where the spermatic cord eventually terminates, and then you have the vas deferens exiting here. And you can see it kind of makes this sharp turn. It initially enters into the abdomen. You can see here's the femoral artery and the femoral vein, which will be entering into the leg as they exit the abdomen from the external iliac arteries here, which are coming off the common iliacs, which are coming off the bifurcation of the aorta in the abdomen. So you can see we're right here, just to give you some orientation, right here in the abdomen. And then you have the vas deferens enter into the abdomen and then sharply turn downward and enter into the pelvis. Now, when it gets into the pelvis, it actually travels along the bladder here. And you can see this here from the sagittal view, and then this posterior view here, you can see where it actually kind of wraps around the ureter. Now remember, the ureters are coming down from the kidney and carrying the urine where it could be stored in the bladder. And in the male, remember, the prostate gland is just inferior to the bladder. It actually contains what's called the prostatic urethra, because remember, in the male, the urethra has several different parts. It's much longer than in the female, and so one of those parts is the prostatic urethra, which is very important in this lecture, and we'll talk about here in a second. So you have where the vas comes in, or the a vas deferens comes in and then actually kind of widens out. You can see this here where it has this caliber here, and then it eventually widens out in this terminal portion called the ampulla, which is where then eventually it merges with the ejaculatory duct, with the secretory duct from the seminal vesicle, which is one of the accessory glands or secretory glands of the male reproductive tract. So this summary statement kind of summarizes the path of the vas deferens. It travels through the spermatic cord, travels into the abdomen, and then it travels into the pelvis, over the ureter, and then terminates over the posterior aspect of the bladder into the prostate. And you can see that here. So like I said, the terminal portion is called the ampulla. You can see that here. It's actually labeled for you. So this is a cutaway of this previous diagram here. You can see we just kind of took the bladder out and focused in on showing the inner portions of these structures, the vas deferens here, the seminal vesicle here, and then the prostate gland with the prostatic urethra here. And like I said, it's wider with a larger lumen in this ampulla portion. And then the ampulla, like I was saying, it converges with the secretory duct from the seminal vesicle to form the ejaculatory duct. So you can actually see where the vas comes in here. It's terminating here. So here's where it's merging with the secretory duct here. And again, just to label this for you, this is the seminal vesicle. And so the seminal vesicle, and we'll talk about its function here in a second, but it secretes its secretions via the secretory duct and then essentially merges with the vas deferens here to form what we see here labeled the ejaculatory duct. So if we actually kind of go up here, here's the vas coming in like this. You merge down here. So you have the vas deferens here. You have the secretory duct here, carrying from the seminal vesicle as V. So the secretions from there coming that way. You have the sperm coming this way, and then they essentially converge at this point right here, which is the ejaculatory duct. Now, the ejaculatory duct contains no muscular layer. It's essentially just a passageway or a conduit, and it courses through the stroma of the prostate gland. So if you look at this, is a posterior view of the prostate. If we're actually to look at a cross section, so if we were to kind of cut across here and look at a cross section of the prostatic urethra actually travels through the center of the prostate gland. So you have the stroma like this, and remember you have, the seminal vesicle here on kind of the outer surface. As we show here on this diagram here, see how it's attached to the posterior surface. Same thing with the ductus deferens. So you have the seminal vesicle like this, and then you have the vas coming in like this. And so what they'll do is, they converge like this, and then they travel through the stroma like this. So here's the ejaculatory duct, like I said, taking the contents from both the ductus deferens and the seminal vesicle, and then converging into the prostatic urethra. So that's what we mean by coursing through the stroma. So it takes these contents that converge on this posterior aspect and then bring it in towards the center where the prostatic urethra is. Now, remember that there's a right and a left testicle that each have their own vas deferens carrying sperm from it. So you're going to have a right and a left ejaculatory duct. You also have a right and a left seminal vesicle. So you have both of these converging here, and they connect and open into the prostatic portion of the urethra, like I said, and it's in the prostate gland at a structure called the seminal colliculus. Now, just to kind of blow this section up here to kind of really show you what this means, because this diagram can be a little bit confusing. So if we draw first the prostatic urethra like this, and we have both of these ejaculatory ducts coming in like this, like I said, they're coursing through the prostate tissue like this, and they're coming out here and they're joining from the seminal vesicle and the ductus deferens or DD, and same thing out here. We'll just draw these in here. So everything's converging on the prostatic urethra, which is what we see here. Now there's this indentation, also known as the prostatic utricle, in the posterior aspect, which is this portion right here. And this is actually between the openings. So you see how there's going to be one opening here and then one opening here. So that corresponds to this in the cross section. So you have one opening of the ejaculatory duct here, you have one here, and then in between you have this indentation called the prostatic utricle. You can see it's also labeled here. And then all of this structure is called the urethral crest, which is this portion of the posterior aspect of the prostatic urethra that receives the ejaculatory ducts, and then it also is receiving via very tiny ducts the secretions from the prostate gland. So everything converges here within the prostatic urethra. You have the sperm from the ductus deferens, you have the secretions from the seminal vesicle, both coming via the ejaculatory duct, and then you have the prostatic secretions as well that are converging in this region. And the portion that receives the prostatic secretions is called the prostatic sinus. Again, that may be a little bit beyond a histology course, but in anatomy, you may need to know that for your exam. So let's actually get into the histology here. So this is a great cross-section of a vas deferens. As you can see here, the vast majority of this structure is smooth muscle tissue. Here's that small lining of epithelium here. Here's that folded lumen, narrow folded lumen that we talked about on the first slide. We talked about the vas deferens. And so as you can see, you have these three layers, and we'll show this to you in a second here. So you have the X here marking the inner longitudinal layer. You have the plus sign marking the middle circular layer, and then the star marking the outer longitudinal. Notice the orientation of the fibers. These are coming into and out of the page. So they're like this. We'll draw it like this. They're coming into and out of the page. So this is the inner longitudinal. Then you have the circular, which is like this. And you can see these fibers. Look at them kind of swirly like this, encircling the lumen like this. So these are at 90-degree angles. And then again, you go out here where the star is, and you have the into and out of the page, the outer longitudinal layer. Now, if we zoom in here on the epithelium, again, it's pseudostratified columnar epithelium. You can see that here. It's a very folded lumen. Now, on first glance, when you look at this like this, you may think, well, how do I know this isn't the ureter? Well, it's kind of hard to see at this point because the ureter is also a very muscular tube. The biggest way you're going to be able to tell that is the epithelium. Remember, in histology, again and again, common theme is the epithelium. The epithelium will tell you a lot. And so remember, in the ureter, that's going to be transitional epithelium or urothelium, which lines the urinary tract. You see it in the bladder and the urethra as well. In the vas deferens, you see pseudostratified columnar epithelium. And remember, that's consistent with where it's carrying contents from. It's carrying it from, the ductal system, which, especially the terminal portion, is lined by pseudostratified columnar epithelium. So that's how you can kind of differentiate those. And again, you have this layer of loose connective tissue here. You can see kind of surrounding the epithelium to provide support. And then immediately you transition into first this inner longitudinal layer, and then eventually making your way out to the other layers of the smooth muscle. All right, so that closes out the third of 4 lectures for the male reproductive system. In the fourth lecture, we're going to be talking about the different glands, the seminal vesicle, the prostate gland, and the male urethra.